And hey friends, so Lab 2. What we're going to do this week is part one of a two-parter. And we are going to be exploring the experiment, more or less, that Avery and McLeod and Griffith did back in the 40s. Essentially, the idea that DNA is what they refer to as the transforming factor, something called the transforming principle. We're going to do a transformation. So it works like this with the transforming principle idea, is that there is genetic material in bacteria that can be transferred from one bacterium to another. Specifically, what we're going to do today is something that is known as plasmid isolation. And let's see if we can have a better marker. So a plasmid is what's referred to as an extra chromosomal piece of DNA. The bacterial chromosome, so here is a bacterium, not drawn to scale, and the chromosome is actually just a circle of double-stranded DNA, and it's somewhat anchored to the cell wall, or the inner cell membrane. And then we have plasmids that can be carried in a bacterium. And these are much smaller circular DNAs. And they are floating free. So plasmids contain, in nature, a variety of different kinds of interesting genes. Typically, when we talk about antibiotic resistance, that is a gene that is going to be associated with a plasmid. And so bacteria can transfer plasmids from one to another, what's called horizontal gene transfer, and you're transferring plasmids. Bacterial, sort of in the wild, might have plasmids that are 200,000 nucleotides. When we're working with recombinant DNA, we use plasmids, but they generally are around the size of about 3,000 nucleotides. And we're going to be working with a specific plasmid today. So next week, we're going to take this plasmid and put it into bacteria. This week, we're going to start out taking bacteria with the plasmid, cracking it open, and extracting the DNA. So it's kind of a circle. All right, in terms of the process. So here's the basic principle then. The plasmid is free-floating. And if we crack open the bacteria, like a little egg, crack open the bacteria, we're going to have the chromosomal DNA will be associated with the bacteria debris in the bacteria cell wall, the bacterial membrane cell wall complex. And the plasmid will be floating free. And so there are a number of techniques that we can use to crack open the bacteria, and this actually is part of the nomenclature. One way is to do something called boiling lysis. As the name implies, we lyse open the bacteria by boiling. You boil them up, crack them open, they explode, and the plasmid goes out. The other primary way is something that was referred to as alkaline lysis. And here we're going to use sodium hydroxide to crack open the bacteria. All right, so now we're going to be using a kit from the Promega company. Hold on a second, I'm going to show you some of the bits. I got a fresh kit in today just for us. And this is, in some ways, a bit of a cheat. It makes very pure DNA. It is more or less the way most labs purify DNA these days. And so I thought it'd be worthwhile giving you a little bit of experience in doing sort of industry standard plasmid preps. Promega, Madison, Wisconsin. They're my company. And, all right, so we're going to crack open the bacteria, And then in the old-fashioned way, you would spin down the pellet, the bacterial goo. The plasmid is free-floating. You take that, and then you would process it. One of the things you do is extract it with a mix of phenol and chloroform, which pulls out the protein, and then you precipitate the DNA using ethanol. Phenol and chloroform are a little nasty, so we try not to use them. What we're going to do is take advantage of the fact that DNA is negatively charged, and under high salt conditions, it'll bind to a little plug of an ion exchange material. Just to show you what this looks like. We have what are referred to as a column. It's a little teeny guy. And you see that white thing? That is the plug of ion exchange material that binds to the DNA. So, the protocol, once we do the lysis, is to actually bind the DNA to the membrane. And we do this by centrifuging, spinning down the lysate in this little membrane cup. The DNA binds, and then we're going to do a series of washes. And then we elute the DNA with pure water by lowering the ionic strength. The DNA can no longer bind to the membrane filter, and it flies right on through. So, it's a fairly easy process. And I've posted and given you both the manufacturer's full 35-page document, and then the single-page quick protocol. You can feel free to look at the 35-page document and read all about it, but really all you got to do is pay attention to, I'm looking at it over here on my computer, the pure yield plasma mini prep system instructions. That's what we're going to follow. So we'll have a series of solutions. I don't know that I'm going to be able to aliquot them out particularly well, and so we're going to have to just take turns and be thoughtful and get our bacteria done. So it starts out like this. I'm going to grow up tonight bacteria with a plasmid, and the plasmid confers ampicillin resistance, so we grow them in the presence of ampicillin. Tomorrow, you're going to have bacteria, and I'm going to give everybody a little bit, and it works like this. We're going to take the bacterial culture, and they have a cell lysis buffer mix that is essentially a sodium hydroxide material. We add that, mix the samples, and one of the things that you want to be careful is that you're mixing gently, because if you vortex, you can begin shearing the chromosomal DNA and contaminating your plasmid DNA. This principle works because the chromosomal DNA is going to be large and bound to the cell membrane, and so we don't want to go crazy with it. You just want to invert it, mix it by inversion. So you had the lysis buffer, and that's going to crack open the bacteria. Then we add the neutralization solution. Let's see, for the lysis buffer, I'm going to give you the protocol. You can look through it, just have it, I'll even print them out for you. But just so you know, 100 microliters to a 600 microliter culture. For neutralization, it is 350 microliters, so you use a P20, a P1000 for this. And then we're going to use the microcentrifuges, and we've got at least two. I'm probably going to bring a few more in so we don't have to crowd around them. The microcentrifuges, we like to have balance tubes, so you have to take turns or put a balance tube in there, and spin it down at maximum speed for about 3 minutes. So spin for 3 minutes. These are going to be in the little microcentrifuge tubes, since what they call that, because they fit in the microcentrifuge. And what should result is a little blob of bacterial debris, bacterial debris, aka the pellet, and then you're going to have the juice, also known as the supernatant, often abbreviated as the soup. And the plasmid DNA is in the soup. The chromosomal DNA is in the pellet. We want the soup. So using a micropipette and being very careful not to disturb the pellet, you're going to take the supernatant and put it into a spin cup. That is one of these little column things. They call it the mini column, whatever. And you centrifuge that down for 15 seconds, and whoop, all of the fluid will go in. The DNA is captured on the membrane. The only critical thing I'd like for you to keep in mind is, at any point, where is your plasmid DNA? First it's in the soup, now it's on the membrane filter. There's a wash step. We're going to add 200 microliters of an endotoxin removal solution, which used to be an optional solution, but they now include it. Endotoxin is stuff that if you were trying to put this into some kind of animal culture cell, could mess the animal culture cells up, and it's generally small sugar molecules and things, and some lipids, and it's part of what makes bacteria really toxic. We're working with non-toxic bacteria, so you don't have to worry about it. Regardless, we do the endotoxin wash, which is 200 microliters, again for 15-second spin. And then we do the next wash is the 400 microliter column wash solution. And at this point, the DNA is still on the membrane. So you do the column wash solution, then we'll take that little mini column and put it into a fresh 1.5 mil microcentrifuge tube. At this point, we are now ready to elute the DNA, and we do that by eluting in, they say 30 microliters, good for them, 30 microliters of pure water. They give us an elution buffer, whatever you like, it's fine. And you then spin that down. All right, actually, you let it sit for a few moments, for a minute, spin it down, and at that point, your plasmid DNA is going to be in a small 30 microliter volume in the bottom of the tube. All right, so now you have your plasmid. What we're going to do next is actually quantify the DNA. So next week we're going to take this DNA and put it back into bacteria, and we'll talk more about plasmid biology next week. But this week we need to actually, or it's nice to know the actual amount of DNA in micrograms per microliter or micrograms per mil, whatever units. And here's how we quantify DNA. There are a number of ways you can do it, but this is how we're going to do it. We're going to take advantage of the fact that DNA absorbs UV light in a very characteristic way. So if we look at absorbance, and it's UV light, and so not visible light, and we can use the UV spectrophotometer that we have here in the room. One of the things is it requires about 50 microliters, and so you can dilute your DNA, and that works. But we also have something called a nanodrop spectrophotometer that uses a single microliter, or maybe two, on a little teeny little droplet, and we can measure very effectively there. It's a very cool little toy, so we'll use that. Again, we'll just have to sort of wait in line 6 feet apart, and we will look at the amount of DNA we have. Here's how we get this information, though, is that there are two wavelengths of light, and wavelengths of light is abbreviated lambda. That's the nanometers of light wavelength, and UV light is starting around 300 nanometers. Bigger than that, it's visible light, and you see it as like, you know, dark purple. If you've ever seen a black light, you're seeing the edges of UV to visible light. You can't see real UV light. That's part of what makes it UV. All right. So there's two wavelengths that we measure at 260 nanometers and 280 nanometers. And we do this because DNA absorbs really well at 260 nanometers. And if you were to do a scan, you'd see something that looked like this. So it has an absorbance maximum of around 260. Now, it turns out that protein also absorbs UV light, and particularly the tyrosine amino acid absorbs UV light mostly in the 280 range, but you've got a overlap like this for protein. And actually it's bigger than that. Uh-oh. So what it means is there is actually an interaction between a corresponding absorbance of protein and nucleic acid at 260 nanometers. And so one of the ways we judge the purity of the DNA prep is do a 260 absorbance to 280 absorbance ratio, because the DNA still absorbs at 280. And this is going to be a purity level. It should be around 1.5 or so. So I didn't draw that out well, and I don't have the number on my top of my head. We don't have to worry about it. But we'll do a nanodrop reading, and the machine does the calculation for you. It'll tell you your purity level. So you'll write this down. And then next week, when we take this plasmid DNA and put it back into bacteria, you'll be able to calculate the transformation efficiency, which is calculated as the number of colonies per microgram of DNA. And so we'll be able to do a little bit of calculations and see. And so again, we have a friendly little competition. Who can get the most DNA? If you're keen on getting the most DNA, talk to me and I can tell you how you can cheat, and it works. So that's for lab this week. Working with E. coli, non-toxic, non-pathogenic. Don't drink it, but you can wash your hands afterwards and be perfectly comfortable, safe. And I'll have the Plasmid Mini Prep quick protocol to hand out, but go over it a little bit, see what it looks like, and it should be actually a fairly fast lab. And then next week we're going to do a little bit more involved stuff, putting it back into bacteria. All right, that's all I know, and I'm going to go start some bacteria right now. Thanks a lot, guys. I'll talk to you tomorrow, Thursday, whenever. We'll see you. Bye-bye.